Standing up here, we should be starting anytime now. Those colleagues joining us online, I also express my opinion. We should be starting anytime. 7 minutes.
His Excellency Ambassador Mayan Atif Sharif, Ambassador of Pakistan to Ethiopia, His Excellency Ambassador Jamal Bakr Abdullah, Ambassador of Ethiopia to Pakistan, Professor Iqbal Oldhai, Coordinator General, Comstech, and Director ICC BS University of Karachi, Professor Sanbata Gudeta, Director of Research and Technology Transfer, College of Sciences, Addis Ababa University, Professor Abba Fakadu, Head CDT Africa, invited guests, ladies and gentlemen, all protocols observed. I would like to thank you all for joining us in this important workshop, which focuses on research in Leishmaniasis. As you all know, Leishmaniasis is one of the neglected diseases. Of course, it's a tropical disease afflicting humans. It is the third most important vector-borne disease after malaria and lymphatic filariasis. It has become a common disease in all over the world currently. More than 1 billion people live in areas endemic for Leishmaniasis. An estimated 30,000 new cases of visceral Leishmaniasis and more than 1 million new cases of cutaneous Leishmaniasis occur annually. Visceral Leishmaniasis, as you know, is a poverty-related disease affecting people, especially living in rural areas. Several studies have been conducted in an attempt to come up with better diagnostic tools and treatment strategies, in particular for visceral Leishmaniasis. A very good example is the one which has been conducted by LEAP with the support of Drugs for Neglected Disease Initiative, but we still need more studies in this area as it still remains neglected. The current workshop focuses on hands-on training in Leishmaniasis research. Of course, PhD students, postdocs work on Leishmaniasis will be the one who will be involved in this training workshop. Leishmaniasis, as you know, is a very important, which needs special focus, especially for those people living in tropical areas, including Ethiopia. Let me briefly introduce the program of the morning session. First, Professor Abba Fakadu, Head CDT Africa, will welcome our audience, followed by the opening remarks given by Professor Sanbata Gudeta, Director of Research and Technology Transfer, College of Sciences, Addis Ababa University. Then His Excellency Ambassador Jamal Bakr Abdullah, Ambassador of Ethiopia to Pakistan, will give us the first keynote address, followed by the second keynote address of His Excellency Ambassador Mayan Atif Sharif, Ambassador of Pakistan to Ethiopia. Before we break for tea or coffee, we'll have a group photo. Just after break then, we'll proceed with 3 presentations, followed with a brief discussion. All the presentations focus on Leishmaniasis. Then we'll have lunch together. The afternoon session will be on battery workshop for hands-on training at College of Health Sciences within the premise of the Kurambessa Specialized Hospital. Now, let me call upon Professor Awa Fakadu, Head CDT Africa, to welcome our respected audience. Prof. Awa, the floor is yours.
Morning again, everyone. Hope you can hear me. Colleagues joining us virtually. Good morning. I do apologize again for starting a bit later about 10 minutes. Your Excellency, Ambassador Sharif, Ambassador of Pakistan, thanks for joining us. Online, we have Professor Badri, Coordinator General, Comstech, International Professor and Director of ITCD, Karachi. This is the Director of Research and Technology Transfer of the Political Sciences, Sabah University. Colleagues from Cape, colleagues from Stanford, the other colleagues. Okay. Neglected tropical diseases, as was just mentioned, affect over 1 billion people globally, the poorest and most marginalized billion. In fact, this is actually an old figure, and the number with increase in population would be much, much bigger. Leishmaniasis is, in fact, the most lethal and expanding NTD. Eliminating Leishmaniasis is non-negotiable. But what is required to eliminate the Leishmaniasis elimination agenda? And let me mention 3 things that would be required. First, improving access to available care. Second, enhancing detection of disease and prevention. Third, making new, safer, and more effective treatments available. Of course, Leishmaniasis cannot be eliminated by itself. The broader intervention to eliminate Leishmaniasis would also have an impact on the broader elimination of NTDs. So in terms of these 3, the truth is that we're well behind schedule in all these 3 fronts, despite the effort that has been going on for 30, 40 years. First, improving access to care requires integration of NTD care into primary health care. In Ethiopia, definitely this is a prioritized and leading principle in all healthcare conditions. And we are also, and what's essential is building the evidence, and we are trying to build the evidence, and more work is required to enhance the uptake of evidence into practice. And I could give several examples of this. Second, enhancing detection and prevention. Better point-of-care diagnostics are definitely required. Current diagnostics are either invasive and have inadequate sensitivity and specificity. Diagnostics for cutaneous leishmaniasis is even poorer, and vaccine development is far behind. Thus, more needs to be done, and more urgently. And third, improving treatment. Safer, more effective, and more affordable treatments are necessary. And resources for developing new treatments are in short supply. The expertise and infrastructure in countries where the burden of disease is the highest is also extremely low. And I think this is where this training and this collaboration is critical. And my heartfelt gratitude goes to Professor Iqbal for engaging with us in good faith partnership so far. And it has been an eye-opener for me to learn about the International Center for Chemical and Biological Sciences. And it does, as a pioneer institution, all the things CDT Africa is aspiring to do. Thanks also for colleagues from
Durham University for your global perspective and engagement. And many thanks for Comstech for making this possible. And I can confirm with full enthusiasm the commitment of CDT Africa and Addis Ababa University to work with you and to do its part to ensure our joint efforts are successful in a short time. And I cannot express my sense of urgency in simple words, but the field of medical discovery has been new for me. Sound negative, but a lot could be done, but a lot is not being done. And it rests upon us, upon the young generation, to do something about this. And something could be done, transformative could be done the next 10 years if we join hands as we are trying to do. CDT Africa is a new challenge-driven regional center of excellence. It has no other priority than building medical discovery capabilities in Africa for diseases of the region. Its second ambition, as it's based in Africa's capital, is to serve as a global medical discovery hub. And this, I would very much welcome our partnership with Durham University, with University of Karachi, and with Comstech as well. CDT Africa is honored and privileged to join hands to facilitate this workshop. I'd like to thank Dr. Fozia, who boldly actually she was quite instrumental. She's one of our postdoc fellows who visited Kenya, and she was instrumental in initiating this program. And she remains a catalyst for this workshop. And finally, on behalf of CDT Africa, my colleagues, I would like to thank you all for joining us this morning and welcome you to this workshop. Thank you so much.
Thank you, Prof. Ababa, for the welcoming speech. Could you please join me to give a round of applause to Dr. Ababa for his wonderful speech? Thank you. May I Sambeta Gugleta, Director of Research and Technology Transfer, College of Health Sciences, to give us an opening speech. Professor Sambeta, the floor is yours. Thank you.
Thank you very much, Professor Yasu. And let's Mark. And if by any chance, excuse me, His Excellency, Mr. Ambassador Atif Sharif Mayan, Ambassador of Pakistan to Ethiopia, Professor Iqbal Chowdhury, Coordinator General, Comstech Distinguished National Professor and Director, ICCBS, University of Karachi, Professor Ababa Fkadu, Head of CDT Africa, Excellencies, ladies and gentlemen, all protocols. I'm pleased to be part of this important launching event, representing myself, the College of Health Science, the University Research Office. As you might well know, Sabah University is the largest and the oldest university in Ethiopia. It has 14 campuses, and the university runs 70 undergraduate and 209 postgraduate programs, and it hosts almost close to 50,000 students. Addis Ababa University is the first in Ethiopia to be an autonomous university. Opportunities for conducting high-impact research, expand partnerships, and run educational programs guided by the needs of the students and the nation. The College of Health Science and this includes tertiary cancer referral. I can see familiar faces who have been working on Leishmaniasis, like Professor Asrat in expansion of early the International Center for Chemical and Biological Sciences. I'm also equally pleased that we are extending this. And gentlemen, I would like to take this opportunity to acknowledge the role of and since its establishment, the center has modeled commitment and high research productivity. And I'm eager for this new partnership to support CDT Africa in its effort to chart an effective path for medical discovery, research, and product development of Africa. Ladies and gentlemen, for this tripartite partnership between University of Karachi, Durham University, and Addis Ababa University to yield enhanced results, I encourage you to keep the longer-term goal of eliminating leishmaniasis from our world. That's a critical ambition that will be rewarded if you also keep longer-term perspectives in a framework of equitable partnership between the different parties. In this process, I would like to assure you the full commitment of our university, that's at Addis University, to support the partnership and the end goal. I wish you a fruitful deliberation, and I declare the conference officially open. Thank you so much.
Thank you, Professor Simbata. May I ask the audience again to join me to appreciate Professor Simbata. Thank you all. Now it is for keynote address. I would like to call upon His Excellency Ambassador Mayan Atif Sharif to give us his keynote address.
Professor Ababa Ficado, Professor Sinbata Guteta, honorable guests from University of Durham, Professor Iqbal Chaudhry, Coordinator General Comstech. Ladies and gentlemen, good morning. It's an honor and a privilege to be here to talk on this important healthcare issue, which is affecting such a vast swaths of humanity. Leishmaniasis is what I've read about this thing is a tropical, subtropical disease affecting more than 19 countries and a large a large proportion of population, affecting mostly the poor people who are malnourished, and can cause loss of livelihood and economic losses for the families. And the care treatment I've understood is very expensive. Therefore, there's a need for enhanced research into the diagnostics, treatment, and care for the leishmaniasis disease. It is for this reason that Comstex and ADT has come together, joined their hands to have this event and to have more collaboration in research and for the diagnosis and treatment of the disease. As well as University of Durham is participating, University of Karachi is also making a contribution. So I really laud the good work of all these institutions. And there's as I was discussing with my colleagues from UK, that there's a need for North and South cooperation and South-South cooperation on these healthcare issues, because COVID has also given us an idea that The global health system is very fragile and very much connected, and we need capacities in all parts of the world, and not only in the advanced world, but in other parts of the world. So we are happy to note that Comstech has brought together, engaged 3 experts from UK as well as Pakistan in this event, and this is the third of the series of events, which the 2 previous were held in Kenya in the last 2 years. This is focusing on the neglected tropical diseases. So Comstech's mandate is to promote the development of science and technology in the OIC member states. Just a few words about the Comstech. It was established in 1981 after the OIC resolution in the summit in Makkah Mukarramah, under the summit. And Pakistan is hosting this Comstech Secretariat. It's also part of our efforts of science diplomacy. And Pakistan takes this organization very seriously and utilizes all the available resources to promote the establishment and promotion of science and technology. Comstech has taken a number of steps apart from this one to promote science and technology. They include establishment of consortiums of excellence. These are the best research institutes in OIC member states. It also includes 500 scholarships for the students of Palestine, research fellowships for many countries. It also includes capacity building in healthcare, eye treatment, in halal products, in pharmaceutical thing. It's training for the technicians, for the laboratory, for the testing. It also includes a science portal to promote science diplomacy, to promote technology actually. It also has various other initiatives, like in similar initiatives, distinguished scholarships programs, and we are we will. And the good thing about Comstack is that within Muslim world, there could be some issues, but or divisions or differences, but Comstack is apolitical and non-ideological, so it brings all the OIC members on one platform, and many OIC members are part of Africa. So we are also happy that through this we are engaging with Africa and beyond, just not just the OIC members, but also non-OIC members, because issues are similar. We are in one boat. Whether we call it OIC or anything else, health issues are common to all. And the good thing about Comstack is that it is also collaborating beyond OIC with different institutions and organizations. Now I will leave more detailed remarks to Professor Iqbal Chaudhary, who is the the expert and the professional in this field. Thank you very much for inviting me again. I wish you all the best and success for the endeavors.
Thank you, His Excellency, Mr. Ambassador. I know how busy you are, and I would like to ask you all to appreciate his time. He has just given us his time from the busy schedule which he has. So I think he deserves a round of applause. Thank you once again, Mr. Ambassador. Then the next address would be by Professor Dr. Iqbal, who is Coordinator General Comstech and Rector. He's following us online virtually. So the floor is yours, Professor Iqbal.
Thank you very much, Excellencies, particularly His Excellency Mr. Jamal Bakr Abdullah, the Ambassador of the Federal Republic of Ethiopia, His Excellency Mr. Mia Atif Sharif, the Ambassador of Islamic Republic of Pakistan to Ethiopia, Professor Dr. Abiba Fikado, my dear friend, also my dear friends and leading experts in the field of drug discovery and development against neglected tropical diseases from Durham University, Professor Dr. Paul Lenny and Professor Dr. M.K. Paul, the Rector of Research, Professor Sinbata Kuteta, Professor Eyasu Mokanen, ladies and gentlemen, leading experts, Assalamu alaikum, and very good morning. I welcome you all to this very comprehensive and inclusive event organized jointly by Comstech, GCRF, NTD, CDT Africa at the Addis Ababa University, entitled Training Workshop on Leishmaniasis. Ladies and gentlemen, I was looking forward to be with you, but because of some technical reason, I wasn't able to travel at the last moment. Fazima, can you hear me?
Yes, sir, we can hear you.
Okay. My relationship and productive collaboration with Addis Ababa University spread over 30 years. When I started traveling to Addis Ababa, where I used to have my good friends, Professor Abigaz, Burhano Abigaz, and Professor, a large number of faculty members have been very close to me, and I was able to travel to your wonderful country and university, and was very impressed right from the beginning, the quality of work being conducted. My point of contact was Professor Ermias Dagne, a very renowned chemist from your university, and I'm very pleased that the now new generation of scientists, including Professor Ficado and his team, is working closely with us on comstech program and the program of International Center for Chemical and Biological Sciences. The idea of organizing these events in Africa came from a discussion which was held in Durham during our last visit, which was, I believe, was in 1921, and that we have decided to focus on Africa and to organize such capacity building events in various countries of Africa. So we already had events in Kenya. Now we have this very important event, which is the result of a good partnership which is emerging out of our discussion with CDT, the Center for Drug Discovery and Therapeutics, a premier institution of Addis Ababa University with a world-renowned prestige and credibility. And then we intend to go to West Africa. We are in the process of developing partnership with the institution in Benin, Gambia, and Ghana. And I'm sure that we would continue to have support from the Durham University. Our ambassador, His Excellency Mia Atif Sharif, has already introduced you to Comstech. Comstech is an OIC Ministerial Standing Committee for Science and Technology Cooperation in 57 countries in 4 continents. It represents over 2 billion people of these 57 countries, and OIC, as you know, is the second largest organization, intergovernmental organization after United Nations. Comstech, as he has very rightly mentioned, go beyond this religious and ideological boundaries and work together with many, many countries, including institutions in non-OIC member state. We have had a very productive collaboration in the past, and we have now a very active partnership with institutions like International Foundation of Science, in Sweden, International Center for Genetic Engineering and Biotechnology, in Trieste, and NTD Africa, the World Academy of Sciences, which I happen to be the vice president and fellow, and also with UNESCO and other UN organizations. With Ethiopia, we consider that an extremely important partnership. Ethiopia is certainly leading Africa in many ways, being the headquarters of African Union. It provides that kind of an ambience and leadership which can have a huge impact on the entire continent. We also know that Ethiopia is spending a lot of money and efforts in improving the social indicator, including healthcare. But it is one country which is affected from neglected tropical diseases. And I don't need to explain to you that neglected tropical diseases are neglected by all definition. They are neglected because they are not part of the global healthcare agenda, and very little money is available for research and development. Ethiopia has, out of 20 declared NTD, 16 neglected tropical diseases are reported from Ethiopia, and a very large burden of Leishmaniasis, which is the focus of this particular workshop. Now, we know that out of one workshop, we'll not be able to develop any blockbuster drug, but the objective of this event is to develop partnership. We develop partnership that eventually lead to better understanding of the disease for the prevention, diagnosis, and treatment. And of course, to have interest develop of young people so they start working in this particular field. If we keep waiting that pharmaceutical companies will develop vaccines and therapeutics, then it's a long haul because there is a little interest in this particular field. I would not really take much of your time. Would just like to add on of what our ambassador, His Excellency, has mentioned about comstech activity. Comstech is the most eminent and active organization of OIC system, as has been mentioned during the coordination meeting of OIC institution. By virtue of its partnership with large number of international organizations and through its numerous programs in Africa and other part of the world. In Africa, we are active in developing institutions, so we were able to develop a high quality ICT master program and the facilities required for such program with the UAT, University of Engineering and Technology, Lahore, with the help of Islamic Development Bank. We are also in the process of establishing Faculty of Engineering and Technology in N'Djamena University, Chad. We have already established one citrus processing unit with our premier institution of Arid Agriculture University, Rawalpindi, and funded by Islamic Development Bank. We are also in the process of establishing many other institutions, centers of excellence, and strengthening the existing centers through various ways and means, including capacity building. Ambassador has already mentioned Health Africa program. So we have been very, very active in terms of developing the capacity, not only in various fields of healthcare, but also to have a physical presence. So we have been through ICAMs. We have been in Chad, Niger, Uganda, and also through other activities. We are bringing about 700 scientists from different countries, including Africa, to various institutions in comstech Consortium of Excellence. Our focus is on developing capacity of the youth, because we consider youth as asset for all countries. Ethiopia being one of the largest countries in Africa, among the most populous countries, 13 in the world, has a progressive outlook, and we consider Ethiopia as a very important partner. We're also fortunate to have a very active ambassador of Ethiopia in Islamabad, and he has been one of the most important advocates of Ethiopian-Pakistan collaboration in the field of science, technology, education, and trade. I'd like to end my comments by thanking our dear friends from Durham University. They are spiritually motivated, and they are those souls which really are global citizens. They are the ones who really work for the betterment of humanity and take all pain and efforts which are required to bring goodness and the knowledge which can benefit for the world. Professor Dr. Paul Denny, Professor M.K. Paul, and many scientists in this network from 6 countries. We're also very grateful to my colleague, Khazima Muazzam, who has been instrumental in putting this very good program together. My very dear colleague, Professor Dr. Samar Yusuf, who is a very active researcher in this field and also globally connected to the community who is working on leishmaniasis, particularly young people. We are grateful to Dr. Fazia, who has not only becoming a bridge between the two institutions, but also has visited the International Center for Chemical and Biological Sciences in Karachi, Pakistan. And we found her representative of a high quality youth of Ethiopia. We are grateful to CDT Africa under the leadership of Professor Ficado. This is a premier institution. We are looking forward to have this institution becoming part of Comstech Consortium of Excellence. We already signed an MOU and we are looking forward to have their visit to Pakistan. So we should be able to not only show what this country represents, but also to bring them on board to a larger network of 57 countries. Addis Ababa University, as I mentioned, is a premier institution, and we have had always a very good relationship with Addis Ababa University. We have been very fortunate to have many young people who came to Pakistan. But let's move it forward. Let's broaden this collaboration for the benefit of a larger humanity, the community of people who are affected from neglected tropical diseases. Let's discuss this important disease and develop better understanding, partnership for future. I would like to thank all of you for your patient hearing. Thank you very much.
So you have been instrumental in this partnership. Had it not been for your unreserved effort, this wouldn't have been materialized. So would like to thank you very, very much. Please, the audience, join me to give a round of applause to Professor Fazia. So before we go for group photo, Mrs. Hazima, the program director of Comstech, has something to say. Hazima, it's your floor.
Thank you very much, Professor Yusuf. On behalf of Comstech, I would like to thank His Excellency the Ambassador for gracing us with his presence and giving us his valuable time. I'm also extremely grateful to the audience who is here in person from Ethiopia for your time and for your participation, and also to Professor Dr. Iqbal Chaudhry for conceiving this this capacity building initiative, and of course our speakers from Durham University for their time and for being here with us for giving out this training as resource persons. As we know, Professor Iqbal is not here. So on behalf of Comstech, I would like to present souvenirs to His Excellency Ambassador Mia Sharif for his time here. Sir, please. I would also like to present a souvenir to Professor Dr. Fikadu for his efforts for making this event a reality. Professor Fikadu.
Thanks so much for the gifts which you have given us. Highly appreciated. So now, I think we are in time. We'll go for a group photo before we break for coffee or tea. I'm not sure who has organized that. Emlet, are you the one? Where shall we get for the group photo? Outside. Okay. May I ask all of you to get out so that we can have a very beautiful group photo? Thanks. Who is well known by almost all of us. He's an eminent scientist in the field of visceral leishmaniasis in particular, in leishmaniasis in general as well, in other tropical diseases too. It's good to have you, Professor Asrat. Share your rich experience with the audience. And let me call you then. Come to the floor and share your rich experience to us. Thank you.
Thank you so much, Professor Yasu. I take this opportunity to thank you all for coming here and for organizing this session. The upcoming partnership with you, with Pakistan and Durham, and so that triangle, that is. Thank you so much. So in this session, we will not be talking about hard science, so it's just really conveying information. And so we want to do it softly. So I will talk about leishmaniasis and the basics and issues that are pertinent to, sub-Saharan Africa and also. Right. So, I mean, this is what everybody knows. So on this slide, that disease is transmitted by sandflies, and it's a global problem. So we have, it's a complex disease. We have a number of hosts involved in the transmission, in the life cycle. So there are 20 species of minimum, and we have about 90 species of sandflies that transmit the disease. And undetermined in transmission. So it's really complicated scenario concerning the epidemiology. And I just given the example of the sandfly on the left, fully engorged. And in this particular case, just depicting the hyraxis, the rock hyraxis here, transmitting at least one species, but possibly two species. Again, the spectrum of disease is complicated. So you can see on the left is with the abdomen, it's the small kids affected by leishmaniasis. These are patients from Southeast Ethiopia. All the patients are from Ethiopia. And the usual common disease is not shown here, which is the speckle lesion, And but here is depicted complex patterns of disease in the middle with multiple lesions, a girl of 12 years old. And so it's metastatic lesions all over in the face. And so this girl was never able to really smile. So it's all over. And at the bottom right, you see big nodules full of parasites. And this is also from southeast part of Ethiopia. And then we have on top two, which is not common, but you see more is the skin lymphatics involved, and ultimately this might end up being lymphoedema, and so it's whole spectrum of diseases shown also. It's just to show you how complex this disease is. And of course, those patients are from Leishmania ethiopica. This one is Donovan. Globally, there are few countries which contribute to the burden. So 10 countries, just 90% of the cases. So you have, the green shows the sub-Saharan African countries, and at the moment, this number 50 to 90,000 is probably less because of the elimination that was ongoing in Southeast Asia. So probably the maximum is 50,000 or so, and most of it is from Africa, about 40,000 cases from Africa. And we have cutaneous leishmaniasis, up to 1 million cases annually, and the majority are the Americas, Central and South America. We have the small variants. We have mucocutaneous and diffuse cutaneous leishmaniasis. This occurs only in 4 countries, and Ethiopia is one of them. And we have PKDL, problem in the Indian subcontinent, also Eastern Africa. But within Eastern Africa, it's largely Sudan, South Sudan. And then HIV co-infection is, the comorbidity is it's common in Brazil, northern part of Ethiopia, but also India. So we have all this spectrum. And of course, map shows you, it's a heat map where you get, the highly affected countries in brown color. And this is cutaneous leishmaniasis. Of visceral also likewise, but you can see still the India, and of course the Eastern Africa countries are highly affected. So this would change now. So the colors in India would turn perhaps into, grayer or something like that. The numbers are very less than 500 or so at the moment, from 90,000 to less than 900 or so. Cutaneous leishmaniasis, you can really identify a few high burden countries within Africa. So Algeria being with the highest incidence, and we have Tunisia, Morocco, Libya, and then that's North Africa. But Sub-Sahara, it's Burkina Faso, Egypt, Ethiopia, and of course Sudan also contributes to the burden. So that is in North Africa, 3 species causing it, but largely because of L major, but rarely they also have Leishmania tropica, but also Leishmania infantum, which is usually the cause of visceral leishmaniasis, causing cutaneous leishmaniasis in that part of the region. So we mentioned about the 90 species of sandflies that transmit the disease. You can see some of them here for L major, Papacy, and infantum, quite a few species for Tropica sargenti. And we see rodents, the dog, in the case of Leishmania infantum, and also even Tropica, where you have Gundi, and so on. So it's really complex in terms of the number of hosts involved, and the list goes. This is in Central and Western Africa. Again, 3 species, rodents usually being the host, and we have Duboski and Papatasi, serving as vectors. Eastern Africa, we have 3 species. In terms of burden, it would be Leishmania ethiopica, and it's found male Ethiopia, but we have also isolated foci in Guinea. Major and Tropica are in the region, and they are for Hyraxis transmit ethiopica, but also Tropica. And rodents are known, have been isolated from for the Tropica, and major has been isolated from rodents, and quite a number of them could be PCR positive. So we have for ethiopica and as the sandfly vectors, these are high within Ethiopia. And we have for Tropica sargentii, Severs, and Gagisbergi also only found in Kenya. Leishmania major, Papatasi or Dubowski, so, you know, and so on. So it's really Leishmania tropica and major usually found in the lowlands, whereas Ethiopica is in the highlands. And you know that, of course, Ethiopia, quite a large chunk of the land mass is in the highlands, so it's natural to expect Leishmania ethiopica widespread. Now, visceral, we've seen this map. And so what may be relevant is the third, where we have the arrow on the orange, so that is sub-Saharan within East Africa. More elimination efforts. So we see epidemiologically, there are areas where you get it sporadically, and at times, but largely it's a stable endemicity. It's widespread. We believe it is zoonotic, even though there is man-to-man transmission where the burden is high. And the incidence is variable, so low incidence, you know, and then of course you have moderate incidence, high incidence where you have epidemics. So this is within Eastern Africa mainly. And so North Africa, again, with leishmaniasis. So the three species rarely, Tropica also causing visceral leishmaniasis, rarely, but otherwise, usually this is North Africa, the Mediterranean littoral. So you have mostly infantum, but also Donovani, and a different set of vectors, Perniciosus and Alexandri being the vectors, and-fly vectors, Western and Central Africa. Again, Donovani and infantum. And when you look at that, we see a new set of vectors, for instance, Phlebotomus orientalis in that part of Africa, and apparently orientalis is in the eastern part. And we also see what you call Sargentomia. People consider them as vectors. So it's another phlebotomin group. And so researchers in Western Africa have found that Sargentomia may be also playing a role in that section. Eastern Africa, visceral leishmaniasis, you see the map. These are the hotspots. And the belt there, Sudan and North Ethiopia, it's the largest belt where you see a huge burden, but also historic epidemics have occurred of the region. In this case, so it's obviously it's a killing disease. The highest burden is now East Africa. We can expect up to 40,000 cases. And it's a problem of poor communities. It's usually a rural problem. It's not in the urban areas. We haven't documented any visceral leishmaniasis in urban areas so far. It's always rural communities and poor communities, remotely located communities. And you can see that, for instance, within Ethiopia, it's really the periphery. Mostly, so it's the lowlands, you know, marginalized communities, and so on. By the same token, I think it's a similar situation in Kenya and Somalia. And also now the disease is expanding in Eritrea. So it's now, except the central zone, every zone in Eritrea is endemic for visceral leishmaniasis, everywhere. Now, so visceral leishmaniasis in the Eastern Africa region has, we call it ecotypes. So two ecotypes. You can see the circles. So the northern ecotype and the southern ecotype. They have distinct differences in many respects, which I will depict. First of all, genomic differences. You look at parasites, genomics, population genomics, and use any number of markers from full genome sequencing or keyholes, and a number of markers, you would really see the same pattern. And so we have the North Ethiopian and Sudan, which we call as the northern ecotype, and we have the southern one. So southern Ethiopia, Kenya, Somalia, and so on and so on. And then we have India there. So when you draw the evolutionary tree, and so in fact the southern Ethiopian or the southern ecotype seems to be more similar to the Indian type, and the northern one seems to be closer to the Elephantine. So it's sort of geographic isolation of distinct populations. So that's genetic differences. Do we have other differences? Yes. Well, look at the countries, again, southern north, that's geography, that's obvious. The sandflies, again, distinct. Orientalists in the northern Martian, quite distinct. Genotype has multiple genotypes. It's a mixed population of parasites compared to the southern type, where it tends to appear homogeneous. And we have found quite a number of isolates that are similar to the Indian type. And in the past, this was done by isoenzyme characterization. And so we have, for instance, MON37, MON36, which you don't find in the northern ecotype, but you also find them in Vicarial. In the southern, you don't get Vicarial. Rarely, if any, and you find one in 30 years, it's mild self-healing. It's a lot in Sudan. So I have come with a new figure. So 5% of VL cases, but if you look at the literature in Sudan, they tell you it's 50% of the cases after treatment would develop PKDL Sudan. And this is less in northern Ethiopia. It's about 2% or less of VL cases that are treated develop PKDL. So you see distinct differences in terms of genotype, epidemiology, sandflies involved, and also the clinical presentation. And HIV co-infection may not be really an inherent disease. We see a lot of a unique situation. It's a lot in northern part of Ethiopia. Then we have, in terms of the course of disease, so the northern ecotypes, they tend to be more acute. So patients are more sick and progressive in terms of the time course. But in the southern, it's more protracted, so it takes a while before the patients really succumb to the disease. It takes quite a few months, 6 months, or more, and then the patients would succumb to it. Now, this is important. The last part is about response to treatment, and this is complicated. And when we do in the afternoon, we'll be exposing you to the drug screening experiments, and this is relevant. So in this case, you see paromycin and ambisome southern ecotype seems to respond well, whereas not in the northern ecotype. So we cannot just use one strain, but we have to use a set of strains for screening and validate anti-leishmanial activity. So that being the case for paromycin and ambisome, that doesn't hold true for the rest. For instance, antibody based treatments, they should be effective in all ecotypes, in both ecotypes. So for certain drugs, which means then that any product needs to be tested and verified against the northern and the southern ecotypes. Am I stuck? Right. So this is the northern and on this one, about risk factors and ecology and so on. But we know more about those ecotypes. And so in both cases, it's really a rural problem. And of course, it's both zoonosis and anthropogenic. So that's the case. But in the northern ecotype, because it occurs in high incidence, there is also person-to-person transmission. And it's important to know, lastly, that herd immunity is widespread in the southern ecotype. There is a lot of transmission, there is a lot of exposure, and so people really develop immunity, and hence herd immunity is a lot. This is pertinent to people who working on vaccines, because people are naturally immunized, and among exposed, only 10% of the people are developing the disease. So, if you want to validate a vaccine, this will be a major issue. There are a lot of challenges. This is concerning diagnosis. Of course, we all know that we have RK39-based, RDTs, point of care, and we know the point of care is really important. And the RK39 test is an antibody detection test. So to date, there is no antigen detection test, and this is one area where we need to invest. And so antigen detection tests are not available. There are some other tests like LAMP and NALFIA or so. But then they don't seem to fit very well to the TPP for target product profile for the point of care. So you still need to do piece, depending on the type of, some have extractions, others don't. Some do it direct from blood, others do it on serum and so on. Platform has really progressed well, and we still don't have an alternative point of care outside of the antibody detection test systems. And HIV-VL, we have a problem there, of course, because it is an immune compromised situation, and their antibody detection also fails. So nearly 50% of them will have antibodies, but the rest don't. So your RK39 will not be valid. So that's unacceptable. And PKDL is a skin manifestation, and again, that's where we have still rely on clinical criteria. The normal procedures, the skin biopsy, skin scrubs, they don't work well. So people rely mostly on skills, clinical skills. And similarly, CL is in a similar situation. And again, the conventional methods aren't sensitive enough, and because it's a skin, and then it's tricky to also design an antibody detection test, or at least in terms of sensitivity. So there also we have problems. So we have gaps in terms of diagnosis, and this is something which we all need to address. So treatments, another area of challenge. So you see at the bottom the toxicities for all the treatments that we have: injections, cardiotoxicity, hepatotoxicity, pancreatitis, whatnot, hypokalemia, nephrotoxicity, and so on and so on. Ototoxicity in case of romycin. So it's a whole set of problems, and the treatment is not that easy. So, so in terms of safety, but in terms of efficacy, as you have seen it. So if type, you have to use a really high dose. So this variable and so on, and administration, mostly it's IV or IM. So that's another area. And so really there's a need for a new treatment to emerge. It's another challenge. So ease of administration, safety, and duration of treatment obviously are critical factors. And at the moment, WHO has the elimination agenda, and so there is a problem of that strategy, because they don't have a treatment that is easy to administer and it up. So we are limited by availability of treatments, and so we are not sure that the VL elimination would be successful as planned. And we have PKDL relapses, HIV-VL. So there, we don't have any sterile cure, right? So you could cure, you can achieve clinical cure, but the parasites are not eliminated. So at any time they come back. So, and I don't think there's any innovation that has attained really achieved the sterile cure for Leishmaniasis in general, but particularly for PKDL and to deal with relapses and HIV co-infection. And the scheme manifestations, again, there's a whole array of clinical presentations, and including PKDL here. And even though some of the treatments are effective in vitro, and for those patients, we don't achieve any good results, and partly because most of the drugs don't get to the skin. So that's a problem to address, in terms of mode of delivery and so on. So we have the DCL particularly, and this is really an incurable disease phenotype. It's impossible to cure unless there is really a new modality, and there you have really persistence, relapses, and T-cell responses are dampened. So it's a big problem. So the same story with HIV VL co-infection. They present in a similar manner, and in the case of HIV VL, of course, the CD4 counts don't come back. Even when patients are on antiretrovirals, the CD4 count does not come back as long as they have leishmania. So that's another challenge, and which needs to be addressed. So chemotherapy does not make sense in this case, unless and otherwise we aim to really develop a technology for bringing up the CD4 counts. And those patients, co-infected patients, just a few of them, they are super spreaders. They can infect sandflies in thousands. So they play a key role in transmission, and so we cannot undermine the problem of HIV-VL co-infection. And we have also tested them in our lab, some of the isolates. We have one master student who did this, and he had found a tendency for a shift in the IC50 values of isolates, that were recovered from HIV and VL, co-infections. So that could be another problem. So not only that they don't respond, but they also are super spreaders. Okay, and also a compounded problem in that set of patients. So the current treatment, the combination of SSG and PM, it's 17 days, but now we have others. So another combination which could probably be used for a duration of 10 days. And of course, they do reduce morbidity, mortality. That's important. And by just treating, we also can reduce the spread of the disease. And beyond that, the disease need for innovation. So what we really aim at, and we have partners like the NTD who are working hard on this, self-administered oral treatment. And I know at the moment there is one treatment which is under in Ethiopia, so an oral NTD, and that which should be affordable, accessible, and so we believe such a treatment could be used in VL elimination. That's another neglected area. So even the expertise is limited, and control methods are not there. And so, yeah, what tools? But we also think that natural products might also have a role here, and so we are trying to also set up a system to evaluate, the anti-sandfly, activity of some natural products. This one I want to treat is another more complex area. So as you have seen, there's an undetermined number of animal reservoirs, domestic and wild. And so we have domestic zoonosis, there is sylvatic zoonosis, so on, and it's complex relation issues and the like. So people stay away from this domain, and so it's a complicated area. So vaccines is another one. I just mentioned there's one issue which becomes important is herd immunity. For instance, if you go to Southwest Ethiopia, 90% of the people are exposed. If you test them with Leishmaniasis, they will be positive. And if somebody is positive, you don't get the disease again. So there's quite a lot of herd immunity, and so to be questioned in that locality, but in other places, it would possibly be good. In the northern part of Ethiopia, it's a disease associated with migration, so naive people, and those people succumb to it. So in there, the vaccine would be needed, and of course, you would have good markers, biomarkers. Leishmaniasis is mentioned. It's an old one, but it's a good one. So that could be applicable in that scenario. So I'm coming to an end. So some priorities here. Capacity for research. We all talk about it here at CDT Africa. And so we need a program, a training program, for PhD postdocs, specific training package, and so on. So that's really important. And I'm sure CDT Africa is taking that seriously. And we of course, in terms of the disease, the distribution and the like would have to be addressed. But we emphasize on the research and innovation, diagnostics, treatment, and vaccines. And we know the VL elimination strategy has started in Africa. So by 2030, just in a matter of 5-6 years, WHO expects to reduce case fatality rate to less than 1%. And at the moment, case fatality is variable from country to country. And we have seen in Eritrea, it's around 50%. So that they have the task to get it 15 times less and so on in 5 years' time. Right. So these are priorities. This is the last slide, because in the presentation, there was a question mark about why Leishmaniasis. It's all about what I said. High mortality. In the case of visceral Leishmaniasis, there's on to epidemic in Sudan. When the two Sudans were the same, in 10 years, the disease killed more than 100,000. Marginalized communities, children are affected. The high cost of care, diagnostics treatment, we have seen it's cumbersome. So the disease is generally tool deficient. There's limited R&D. So it's one of the most neglected. The economic impact is high. And there's also a lot of comorbidity in association with Leishmaniasis. Cutaneous Leishmaniasis, it's a disfiguring disease, stigmatizing. It does affect our daily life, including married life, high burden. And again, it's epidemic prone in risk groups. Recently, we had an outbreak in Somali region, so the eastern part, which seems to be associated with L. typica for the first time. And it's only among soldiers who were stationed in the jungle, and no civilian got it. So it's a specific risk group, then it can have outbreaks. This could be incurable, so you can imagine what it means. And of course, they will get the disease also inside their houses in the root areas. And so poor living conditions are associated with it. Like VL, it's also a tool deficient, and we have diverse eco-epidemiology. And as a consequence, it seems worthwhile that this venue is addressing this Leishmaniasis in general. Thank you so much.
Thank you, Dr. Asrat. I wouldn't dare summarize what he has mentioned. Just to mention, point out a few key sentences which has mentioned. Looks that Leishmaniasis is very scary these days, in particular in Africa, with special emphasis in East Africa, including Ethiopia. You've seen how the parasites are well distributed throughout Africa and globally as well. So the highest burden, in particular for visceral Leishmaniasis, is shown in the eastern part of Africa. That was the reason why JNDI, under the platform of LEAP, started to conduct trials and attempt to get new drug treatments for visceral Leishmaniasis. Of course, in some cases, we can say it was successful. Thanks to LEAP, currently we have some additions as far as treatment of visceral Leishmaniasis is concerned. So cutaneous Leishmaniasis still remains neglected, but we have at least some promising data. So he has shown us the distribution of the parasites throughout the world. He has mentioned the challenges, both in terms of diagnosis, especially in terms of RK39 availability for the majority of those who are in need, which is one of the RDTs. And he also mentioned about challenges in treatment. In terms of safety, he has mentioned a number of toxic effects with the currently existing drugs, some of which are not tolerable. In terms of efficacy, we've seen, and he has mentioned efficacy could be variable. As a matter of fact, some drugs resistance has already emerged, unfortunately. Not only that, in terms of convenience as well, most of the drugs available for treatment of visceral leishmaniasis are administered parenterally, intramuscular or intravenous. We have probably one or two drugs only which are available for oral purpose. That makes it even more complex. And the duration of treatment is also very, very long, which tires not only the patient, but the family of the patient who is taking care of the patient as well. So all this would add up to make visceral leishmaniasis, as well as cutaneous leishmaniasis, very, very difficult tropical disease. And on top of that, the issue of PKDL, especially in Sudan, that is very, very common, though it might have not been observed so far in Ethiopia. That is also another issue which he has mentioned. Control of sandflies has already mentioned as well. And finally, he said, our main focus area has to be to control and eliminate visceral leishmaniasis, i.e., by prevention, diagnosis, and treatment. Thanks a lot, Professor Asrat, for highlighting the important points. Please join me. Give a round of applause once more. Thank you so much. Then hope Professor Iqbal and others are still online. Can I hear from you, Professor Iqbal? Are you still online? You might have got lost, unfortunately. Hopefully he will be back when his presentation comes into picture. Okay, that's okay. Then Professor Asrat has answered why leishmaniasis. Okay? Why do we need to discuss about leishmaniasis this morning? Sure, we all got the answer. We are all convinced as well. Then the next presentation is presented by Professor Paul Vivas. He is from Durham University, UK. And he's going to tell us about leishmaniasis, drugs, as well as diagnostics, as disease neglected. He's a professor of molecular parasitology. I'm sure if you lend ears, he's going to hear a lot. Floor is yours, Paul, please.
All right, now the technology is sorted out. So many thanks to Professor Esu for that warm welcome, and Professor Azrat for really introducing why leishmaniasis. It remains a critical disease. So just I'm going to really focus on oh yeah. Anyway, I'll carry on. So today, as part of the comstech and CDT Africa event, and I'm very grateful and actually very honoured to remain part of the comstech effort and also be welcomed by CDT Africa and Professor AB and Dr. Fozio have been very hospitable and showing us around yesterday. So I'm going to focus on drug target identification and drug discovery. This really leads into some of the themes, and particularly the practical, that Professor Azrat with me is going to run this afternoon. So just by means of introduction, I'm in the Department of Biosciences, but I work very closely with physical sciences colleagues in the biomolecular interactions group within the chemistry department at Durham University. Now, this is Durham. It is a beautiful, very small city. There are more students than residents, it seems, most of the year. But that's the famous Norman Cathedral, which is approaching 1,000 years old. It is, if you ever come to the northeast of England, Durham is about 15 miles south of Newcastle. That's the one with the Premier League football team. That's the castle, which is actually student residence at Durham. Beyond this medieval grandeur, we actually work, my colleague Professor Emker and I, particularly in a global context. And as has been mentioned in the introductions and is on the poster there, we are part of the Global Network for Neglected Tropical Diseases. And that includes our colleagues, our valued colleagues in Pakistan, but also colleagues in India and South America as well. So we have a global view. Okay, so my interest over maybe 30 years has been on lipid biosynthesis in protozoan and parasites. A fairly unusual passage and decision to undertake, and it doesn't come without tears, I can tell you that much. But for those of you that don't know, sphingolipids are important lipids in the plasma membrane of eukaryotic organisms, including protozoa. They're almost magic lipids. If you listen to the claims, they're involved in almost everything. But we're going to consider the enzymes involved as drug targets. So whilst my passion lies with Leishmaniasis, I do also work in toxoplasma, which is an important human and veterinary pathogen and is related to Plasmodium, the malaria pathogen. So you can see it's complex at the apex. It's common definitive host, the cat. Its effects on the brains of unborn children and indeed unborn, particularly lambs. And it's related to Plasmodium, as I mentioned. But really, I'm going to use this to introduce the kind of work we're doing, both Emker and I at Durham. So the first enzyme in sphingolipid biosynthesis is called serine palmityl transferase. And biochemically, I'm not going to go into detail. Essentially, it condenses serine and palmityl OA to produce, at the expense of carbon dioxide, this keto dihydrosphingosine, or KDS, which is the base unit of all sphingolipids. So very important. Now, we've known for many years, and I think it was about 2 years ago, wasn't Emker, the cryo-EM structure of the human enzyme in its dimeric form became known as 2 subunits. This is in all eukaryotes, apart from one group, which I'll talk about, LCB1 and LCB2. LCB2 is the catalytic domain. LCB1 is thought to be regulatory, but it's different. Now, toxoplasma, we identified a potential ortholog, and indeed, we have expressed it. We can see its functionality in vitro is the functionality we expect. This is mass spectrometry showing KDS formed. We know it's like the human and other eukaryotic SPTs localized in the endoplasmic reticulum. This is what this shows. But I won't go into this because you can't even see this, but it has a very strange sequence. Now, because it resembles the bacterial autolog, which has a very different evolutionary origin. So the bacterial SPT is a homodimer. It does not have two different subunits. It's soluble. It's not in the ER, of course. And when we looked again at the toxoplasma, again, it forms a homodimer. And here it is modeled with small angle X-ray scattering, which EMC is very involved in. Okay, so it's bacterial-like. So this is how we approach our drug target selection in general. We're looking for differences between the human, essentially the host or animal, we're thinking veterinary, and the parasite. So ceramide is the sphingosine, which you can see here, and it's acylated essentially. So that is a bioactive molecule. That is the building block of, for example, sphingomyelin. That's about 10% of all our plasma membranes in all our cells. And this lipid here, inositol phosphoryl ceramide, which is not a lipid found in mammals. It's found in yeast, plants, and many protozoa. And it has an inositol group as opposed to the sphingomyelin choline group of the phosphorylated group on this particular lipid. Okay, and also, and again, this is a really prelude to some of what Emeka is going to talk about tomorrow. So, is it tomorrow or Wednesday? Tomorrow. Tomorrow is Wednesday. Okay, here on the top left, we have, everyone probably here has heard of AlphaFold, or at least the process of predicting protein structure. So we have looked at ceramide synthase, which is the next step, the next major step in sphingolipid biosynthesis. And we found two enzymes, one of which we've shown to be catalytically active, and the other one is catalytically inactive, because it lacks certain residues in the predicting active site. So with EMCA, we have modeled these, and you see they're basically the same structure, but one is active, one is not. Probably more interesting is the inactive one is indispensable, but the active one can be knocked out. So this is the kind of approaches we're taking. It's working. Yeah. All right, finally, Leishmaniasis. So I don't need to cover this because Professor Azrat has covered most of this in the previous talk, but it remains a major global problem, including, as you can see, in Ethiopia, where he did mention the co-infection with HIV being a major issue. And also, when I went to India, we were both in India, weren't we? Was it 2019, I think? It's just pre-pandemic, early 2020. And the hotel I was in just put a copy of the Hindu under the door. And this was an article that I managed to find. And Kala Azar, there is a elimination effort, as was described, but still causes major problems. This is a teenage boy with that disease. And I'm glad to say also there is WHO and governmental action to try and eliminate these kind of diseases. They are somewhere near the top of some political agendas, but certainly not at the top. Right. So I think what cemented my sort of more translational interest in Leishmaniasis was a visit again in 2019 to the Jinnah Hospital in Karachi, organized by my friend and colleague, Professor Sameh. And I met these children, all caught cutaneous leishmaniasis on a family trip to the northern part of the country, returned back, and they're receiving, frankly, awful treatment, the only treatment by very dedicated medical staff at the Jinnah Hospital. So this really gave my great focus on cutaneous disease and what to do next. So I don't need to go along and through this, apart from to say, as Professor Azrat said, it's a very complex disease, multiple species, multiple vectors, multiple pathologies caused by these species. So there are lots of things for us to consider, lots of things for you all to learn, I hope, in the future. And current treatments are frankly appalling, and the efforts through DNDi, and I think it's the University of Gondar in Ethiopia, to field trials some oral therapies for visceral leishmaniasis are very, very welcome. But I would say the primary therapy for cutaneous disease still remains these very toxic antimonials, and I'll come back to those a little bit later. So we do look at these, and we continue to look at miltefosine, for example, as well as amphotericin B, and particularly miltefosine. It's not an ideal drug by any measure, as Professor Azrat said, but we do need to start to understand how it works, to start to understand how we can make it better and how we can track resistance, which frankly is arriving very quickly. So eukaryotic sphingolipid biosynthesis, I've already mentioned serine palmitoyltransferase. It's conserved, but then we get this divergence for complex sphingolipids. Sphingomyelin produced by sphingomyelin or SM synthase. And then this is what I'm going to focus on. As I mentioned, yeast plants and some protozoa, they produce inositol phosphorol ceramide or IPC produced by this enzyme, IPC synthase. Now remember this, known as OR1 in yeast, because we're going to come back to yeast. Now, why is this a potential drug target? Well, it sits at a cusp. Oh, we've got. Okay. Okay, and apparently there's some technical issues, so we'll be back after this short intermission. Okay, well, I hope everybody can see that, whether they're in the room or not now. So the reason that IPC synthase remains a promising drug target across a range of organisms, or even a herbicide target, is because it sits on the cusp of life and death in a cell, if I put it simply. So its action, which is the utilization of ceramide to produce IPC, reduces the amount of ceramide, which is a pro-apoptotic cytotoxic agent within a cell. And it also increases the quantity of diacylglycerol, which is a side product of the reaction, which is, at least in mammals, is mitogenic. It promotes growth. So it kind of sits in that balance point, the seesaw between cell life and cell death. Also, it's a validated drug target in yeast, particularly this natural product here, aureobacidin, depsipeptide, very complicated, has gone not all the way, but towards the market as an antifungal. But it's the enzyme, IPC synthase, is a complex transmembrane enzyme. There's 6 transmembrane domains, very difficult to think about how you develop an assay to do drug discovery with that. So we've worked on various things, including in vitro translation and peptidoliposomes. But really, the way to get complete traction on this was to use what you could loosely call a synthetic biology approach, and actually replace the OR1 in yeast. And I won't go into detail with this, given the time. I'd use a plasmid shuffle to make this yeast completely dependent on the Leishmania IPC synthase, its functional ortholog. Very different enzyme, but functionally identical. So we have yeast dependent on this. And when you have a yeast, you can actually make a high throughput assay. Okay, so it's a simple cell, it's cheap, it's safe, it's a category 1. So it makes everything a lot simpler. And what we did is, working with GSK at Tres Cantos in Spain, we formatted this into a 1536 well plate. You will never use these in, I hope you never use these in the lab, because you need a robot to actually allocate about 20 nanoliters to each well. So this really is just for you to think about before the practical. This is the kind of maths we used, plate by plate, to make sure each plate was robust. Because if you're screening, in this case, 2 million compounds, you want to know that your assay is robust at a plate level. So this is called a Z factor or Z prime factor. And essentially, it's a mathematical way of looking two controls, the positive, which is a known drug or inhibitor, and a negative, which is a vehicle or no drug or inhibitor. And you work out the standard deviation and the variance between those. And you should have a big gap between the positive and negative, but also small standard deviations. Okay. So we started with 2 million compounds. We worked through a pipeline of screening and then comparative screening between the yeast dependent on the Leishmania enzyme and the yeast depending on its own enzyme. And that's really to sort of get rid of just antifungals. And then we have a dose response, say, which I'll talk about here. And that's about 5,000 compounds at that point. So if you look at this graph here, so it's the inhibition of the OR1 yeast up here and the inhibition of the Leishmania IPC yeast down here. So what we're doing here is dose response, and we're looking for the most selective, and they'll be in this corner here. These are all log values, I should add, but we can talk about that another time. So right in this corner, they don't affect the yeast, or at least not very much, depending on its own enzyme, but they're highly active against the yeast dependent on the Leishmania enzyme. So we had, from that stage, quite quickly actually, gone down from 2 million to 500 in a matter of a month. But then we have to really confirm the action, because there's all sorts of things that can happen in a complex yeast cell in terms of false positives or indeed false negatives. So when we take it into a much more difficult laboratory-based enzyme assay, where we use fluorescent ceramide to look at the enzyme activity in extracts from the same yeast. It is complicated and involves a process I can't possibly go into now, but it does work, but it takes probably about 6 months training for someone to actually be able to do this. And we identified a class of compounds known as benzazepanes and some derivatives which had a sub-micromolar inhibition in the enzyme assay. And of course, we published it. Now, there's some other things. This generated some interest from some of my friends and colleagues around the globe. So we also helped out my colleague Sylvia in USP, University of Sao Paulo. And this is the rather famous anti-cancer drug tamoxifen. And indeed, it appears to have some inhibition of IPC synthase in Leishmania. And this has gone into some small-scale clinical trials in Brazil, actually. And also this, we continue to work with this clemastine fumarate, which came out of a screen that we did with a company, a small company called LifeArt, which are a not-for-profit company in the UK. And we see here work with Bartira Rossi Bergman in Brazil, in Rio. That clemastine actually, at least intralesionally, is pretty similar to the antimonial glucantime. But also we use things such as metabolomics. So we look at the small metabolites that come up and down. This is using a mass spectrometry approach with colleagues at Glasgow. And we have a pattern of metabolites that fit the inhibition of the enzyme. It's never a perfect process because, as I said, the cell is complicated. But we did get the cover of ACS infectious diseases, and I don't know who made that picture, but it wasn't us. So, okay, but I mean, really, I've told you that it's a drug target on the basis of my speculation, but is it really? Because our work and others, we can knock out, we can remove the SPT, we can make cells, Leishmania, that do not make IPC in culture because they don't have ceramide. And indeed, this is work from Kai Zhang, they still actually produce lesions in an animal model, and the same here, this is my stuff. But where does this leave IPC synthase as a target? Well, more recently, we repeated the experiments that were done, 25 years ago, essentially with Leishmania major in the rather more tractable Leishmania mexicana. And we found we couldn't knock out the the SPT catalytic subunit, LCB2, in Leishmania mexicana. It was impossible, unlike Leishmania major. So then we went back to Leishmania major and we actually sequenced the whole genome. And what we found is there's non-targeted deletions. Now, there's a technical process here, and the Leishmania genome is very plastic, which goes in to the ecotropes that were really described in the rise of resistance. Things change. And we actually had a complete clonal loss of this gene, which you can see here compared with the parent. And that's a gene encoding a putative sterol transporter, an ABC transporter. So they were knocked out seemingly simultaneously. And actually, we've used the new gene editing technology to knock them out at the same time in Leishmania mexicana. We can exactly, at that point, recover LCB to SPT negative Leishmania mexicana, but only when we have the sterol transporter lost as well. So it's a complex picture. But where does this leave IPC synthase? So, well, we are working on this, and currently it's in revision. But when we infect mice, we see a very promising decrease, or there's no lesion. We have a considerable decrease in the number of parasites found in the lesion site, or the injection site. But when we try and add back the gene, we have to complement it to try and reverse the phenotype. We cannot. We get the same phenotype. Complex, but not unknown. And again, you know, we can see here, this is quite a simple one. I'll just focus on this. So this is mass spectrometry looking at IPC in these cells. So this is the wild type or parental. You see, we knock it out and all the IPC is gone and our pathology is down. And we can see we add back, but we're not adding back. The enzyme is not as active when we reconstitute it's in the cell. So we're getting rather less IPC. So our argument from this is that we are not getting as much of the enzyme. We are not getting as much of the product. So we are not recovering the wild type phenotype. We've also looked at this in the related parasite, Trypanosoma cruzi, with a colleague, Santuza Teixeira in Brazil and in Belo Horizonte, and indeed the same phenotype occurs. Okay, just really for the sake, I know a lot of you are interested in natural products. I have a small footprint in natural products, but we have worked, and we could talk about this informally, looking at various natural products, including or fungal extracts, rather semi-synthetic saponins, and indeed chalcones, which my friend at the back will like to hear. And we also look at such things as Aureobacillum in toxoplasma. It does have quite an effect, and it does, at least in a cell-based assays, appear to be very active. Okay, but what now? Where are we taking this? I've gained a lot of experience, I suppose. I've shed a lot of tears because things in drug discovery don't work normally. So what we've done is kind of taken this and we've approached it, and we approached Tres Cantos, which is the Open Lab Foundation charity funded by GSK, together with our global network for neglected tropical disease, and really take some of the leash box compounds, and they're compounds that GSK identified as being highly active against Leishmania donovani. We want to deconvolute the mode of action. I persuaded them that the best way of deconvoluting was not to use Leishmania donovani, but to use the Leishmania major and mexicana. So we're actually shifting their focus from visceral to cutaneous disease, rather subversively, because they really focused on visceral disease at the time. So they seem to take this well. So it's an international collaboration. There's Santuza I mentioned, and Iqbal, who you'll hear from later, Ashish in India, Carlos in Uruguay, a team in the UK, including Jeremy Mottram in York as well. So I can't really talk about the data because it's slowly coming out. The project's coming to an end, but also we're tied by confidentiality. Those of you who work with industry know that that's the case, but essentially the process is to go through a triage of activity and cross-resistance and the ability to generate resistance lines to get down to a small number of compounds where we have genomic resistance, lab-based genomic resistance. We have omic technologies such as metabolomics, thermoproteomic profiling, which has worked quite well. And we will gain an understanding of the target or potential targets of the drugs. And this is where MQ and I sat down and we thought, what makes a good drug target? And we will deprioritize those that we don't know how they work, because you can't really follow those up, all the way through to confirm function, which is absolute priority, because we need to then do a biochemical assay. And this might surprise you, but down here we deprioritize intrinsically disordered proteins, but also integral membrane proteins like IPC synthase. Now, I said there was a lot of tears. Working with transmembrane proteins is really, really difficult. And it's gives you insight, but it also doesn't really lend itself to the drug discovery process, at least in an industrial sense. So we prioritize those that are soluble, cytosolic, known drug targets maybe, but particularly unknown drug targets. It's stuck. Let me just push that one. Oh, there we go. So, so it's still a high-risk undertaking anyway. So just bear that in mind. With any of the drug discovery efforts, it's always high risk. And I've come to realize that, and Professor Azrat mentioned this, drugs on their own are not the answer. Really, diagnostics are key. And also, as he mentioned, particularly with focusing on visceral disease, where there's been much more investment, there's not the right diagnostic out there for active infection. So this is the one essentially he was talking about. So that's for Donovani, that's a serology one. And then we do have this rapid test, which is a lateral flow test to detect an antigen. So it does detect an actual active infection, but it lacks a lot of clinical data. It's private, so there is an expense, but also it does not distinguish between species. So our learning from this, our learning from when I spoke to clinicians, is they need to know whether it's in Pakistan, in Sindh, whether it's a Leishmania major or Leishmania tropica infection. They're the two species. Leishmania tropica, about 45 to 50% treatment failure rate with antimonials. Chronic infection can last years. Leishmania major, very unpleasant scarring, responds well to antimonials and even self-heals. So they need to know, they need to sort of start to think about different treatment regimes for tropica infections, which are increasing significantly. And just to say, there are people like Jessica here, who's an advocate now for cutaneous leishmaniasis. She, I would say, wealthy tourism in the jungles of Costa Rica, got leishmaniasis, but she's been very open in publicizing treatment and scarring. But this is what I was mentioning. So this is the antimonial treatment. So this is, you'll see sort of, I would imagine, similar treatments in Ethiopia. This is direct injection into lesions, you can see here. Very common in even sort of well-resourced hospitals in Pakistan. But again, the treatment failure for tropica is 45%. So at the World Leash Congress in Colombia in 2022, there was actually a manifesto presented on diagnostics for cutaneous diseases. And there is now an established target product profile, an ideal. LAMP assays, there are problems, and we can talk about that at length in more private conversations. Have a possibility, shall we say. So we've been working on this over the last 3 years, essentially, and our target was to find differences we could exploit between major and tropica so we could get specific and sensitive assays. So we've only done this at the moment on laboratory strains, but working with Professor Sameh and at the ICCBS, we're looking at clinical strains probably in the winter time this year. So yeah, so we can gain specificity. You can see it here. So that's Leishmania tropica, and we can see down to 1 picogram of Leishmania major, we're still getting a signal. So it's sensitive, it's specific. And I think I want to mention this, and this is where it really comes back to Professor Azrat's talk. And I think we probably could look at north and south ecotropes of Leishmania donovani. There's probably enough difference there in the genome to have differential assays. And to try and make LAMP a assay that can be used in the field, we're working with Marloes Peters research group, and they're essentially medical engineers. So this is to make a low-cost device, a very simple device, where we can add a tube or a series of tubes where the reaction's been taken, and we can get a signal in the field, so without expensive equipment. So I just finished with this. So this is the workshop last year in Nairobi, and we are all stronger together. And I'm losing my connection. And thank you to my group. And before Professor Iqbal gets annoyed with me for overrunning, I think I'll stop there. So thank you very much.
Thank you, Paul. Very wonderful presentations. I would like to congratulate you on what you are doing. I think you are doing a lot of work, both on diagnostic as well as treatment aspects. It's true, you know, when we think of developing a drug against Leishmaniasis, in particular against other parasitic diseases, the major challenges is to get unique site on the parasite, because the parasites are in the host. So when we are targeting to kill the parasite, make sure you don't target to kill the host cell as well. So we need to come up with the unit structure whereby the parasite processes, in which case the drug might be more specific to attack the parasite host cell. That's the major challenge we have. So we have to look into unique enzyme, unique biochemical processes, unique target sites and the likes in order to attack that parasite without affecting much the host cell. That's what Paul and his team are looking for. So that was really wonderful. I would to summarize what you said, more of molecular parasitology, he has mentioned about, which is of course his area. And he has mentioned a lot of issues and a lot of molecules which have been screened by him and his colleagues in his laboratory. And in fact, they were not limited only to treatment, but also to diagnosis. So they also did a lot of work on diagnosis. Let's appreciate him and co-workers. Thank you, Paul. So the next presentation, I'm not sure whether Prof. Iqbal is still online, is he? He's there, okay. Is he muted? No.
Hello, Professor. How are you?
It's okay, okay. Prof. Iqbal,
Yes,
could you give me a signal that you are there?
I'm here very
Let me
much.
make
Can
sure
you
that
hear me?
you are still with us. Yes.
Can you hear me now?
Thank you then. Yeah, we are looking for your interesting topic presentations, okay, on recovery of anti-Leishmanial drugs. So, Mr. Iqbal is going to present on anti-Leishmania drug lead discovery from natural products, which most of us are interested in. As you know, Ethiopia has diversity of different plants, which have got potential to develop lead molecules, become various type of drugs. So let's hear them from him, what he and his colleagues have discovered from natural products. So Professor Iqbal, the floor is yours. You can continue. Thanks.
Thank you very much, Professor, my very dear Professor Ayasu.
Slides are visible.
Yeah, can you
We can
see my
continue.
slide now? And I hope that I'm audible. Can you hear me and see my slides?
I'm not sure whether there is any internet problem. The internet might be unstable. I don't know.
Okay, can
Prof.
you?
Iqbal?
Yes, can you see my slides? Am I loud and clear?
He's still there.
Yes, he is.
Mhm.
His mic is not muted.
Oh, the mic might be problem. I don't know.
Okay. No, we cannot hear him.
Prof. Iqbal,
Yeah.
is there
Can you-
any problem from your side? Maybe from your computer? Your mic may not be working.
Can you hear me? Apparently you can't hear me, right? Oh, okay. Yeah.
We are so eager to hear you, but we're not hearing you. Fortunately. Yeah. Someone is helping him, yeah.
Can you see my slides now, Professor Yasu?
Okay.
Can you see
Hello?
my?
Prof.
Yeah.
What?
Hello.
How about now? It looks the problem persists. I don't know what can do. Huh?
Can you see my slides? Can you hear me? Apparently not.
They're just helping him. I think it could be here. Can you hear him? So it might be from our side then.
Okay.
Yep.
Hello, everyone. Can you hear me?
So it's from here.
We can have him. Yeah.
So you have someone from here who can help us.
Actually, we should hear something.
Hello. I'm trying to call Dr. Samar also. Samar, can you hear me? Can you see my slide?
सर आपकी स्लाइड्स आ रही हैं आप ऑनलाइन सब तो आपकी आवाज आ रही है कि समझ नहीं आ रही
Okay, so the problem is at your end, right?
Is he on? Okay.
It's good.
I'll wait for it.
Wonderful.
Hello, friends. Hello, everyone.
Yes, now we can hear you.
Yeah. It has to be a bit louder.
Okay.
Okay, thank you very much, Yasu, for Can you hear me now?
Just a second, Prof. It's not
Okay.
very loud. Just a second. Apologies.
Wonderful. So apparently everyone can hear me. People...
Prof. Iqbal, just a second. Sorry. Just a
Sure.
second, Prof. We are still
Okay.
having difficulty to hear you. We get your voice. We get a bit of your sound, but it is not as clear as it was previously. Just getting some technical help. This, what's it one? This is not set.
Keep closer to the mic, probably. It could help.
Okay, it's not this one.
Sorry.
ከኛ አይደለ ሊሆን ይችላል። I don't know.
And I think I need to keep talking. So you need to know that, I mean, apparently you need to check your mic from your end, actually.
What happened to your mail? What's happening? It was very clear earlier.
Yeah, presentation.
Vanilla speaker system.
Hello.
Hello, hello, hello.
Hello, everyone.
Yes, we can hear you, but the sound is too low.
Too low.
Can you keep closer to the mic in case that was a problem?
I have a mic right in front of my face.
Yeah, yeah.
Can you hear me now?
We can hear you, but it's not loud enough. That's what I know. I think there is a problem with the audio system. Yeah, yeah.
Apparently, the problem is at your side because I have. And
on that phone.
Yeah, what are they?
hello, everyone.
Yeah, we can go here. We can go here. We can go here.
Please test the
Hello, hello.
Yeah. Hello, brother.
Hello. Could you speak up? Hello, hello, hello. Hello, hello, hello. You try it? Rob Iqbal. Would you try it?
Yes, Professor. Can you hear me now? Can you hear me now? What's been the problem? Hello.
test, test, test. Test.
Hello.
Hello, hello, hello. Yes, can hear you now. Can hear you now.
Can you hear me clearly?
Yes, very good. Yeah. I'm sorry for what has happened. Please continue then.
It's all right. Okay, thank you very much. And my apologies that all these technical problems arise when someone doesn't really travel to the venue. So this is my fault. I take full responsibility. But let me start my presentation. And I must say that I really enjoyed the earlier two presentations. Paul has set the stage for many presentations coming forth, and the one which I'm going to present is shared with my colleague, Professor Dr. Samar Yusuf, who is there with you. And since this is a workshop, I would be discussing some practical aspects of how to get the natural products from complex plant matrix. It is fairly challenging, and for that, I would be discussing medicinal plant and natural products, and certainly various stages in the process of extraction and isolation of natural products from plants, I would briefly be discussing about the neglected tropical diseases, though I would certainly avoid any detailed discussion, and of course, Leishmaniasis, for which we have worked by giving you an example of Phycellus minima and our efforts to understand the mechanism of action. We all understand that plants are extremely important, and they would remain be an extremely important source of very exciting, very interesting, very complex molecules, which synthetic chemists cannot really synthesize despite so much development in the methodology. The medicinal properties of plants are because of secondary metabolites, and these natural products are actually used in our cultures. So they are a very important part of our traditions, culture, and of course, they have a great utility in our lives. So natural products have been the main source of drug discovery and development since literally decades. In between, of course, synthetic chemistry has taken a lot of place, but now people are coming back to natural products, and largely the reason is that the structural diversity which you get from nature is incomparable. And there are so many of these plant-derived medicines which are actually used in the treatment of diseases such as hypertension, cancer, inflammation, and infections. And these are some of the examples of natural products which eventually emerge as drugs. And a lot of them have been used as initiator of further changes in the structures, and a lot more were developed on the basis of the template which nature has given. So nature is and would remain a very important source of structural and chemical diversity, which is required for modern drug discovery and development. And this is the key reason that people keep searching medicinal plants for their chemical diversity, and from traditional natural product till to date, there are a constant flow of new medicines coming out of nature. We have been working in this field since long, and we were able to conduct lots of ethnobotanic surveys and collected lots of information about the use of plants in culture. So this is extremely important for ancient civilization in countries like Ethiopia, for us to understand how people have been using these plants for a variety of different purposes, including in healing practices. And what we have done was that we have collected a lot of these information and put them on record because of two important reasons. One is that we would like them to be gathered for further scientific studies, and second, that we would like these information to be documented so they're not really are biopirated, which is a common problem in the world. So intellectual property right of the practices have been documented, and we have actually conducted surveys in different parts of the country. We were able to develop many drug leads, some of them in various phases of clinical trials, and they were all from medicinal or dietary plants. Of course, traditional knowledge has to be translated into evidence-based medications, and for that, science is extremely important. So you have seen the glimpses of a lot of very interesting science being conducted in Durham University, where natural products are validated for various activities. And this is the reason that our institution in Karachi has developed the range of biological activities. We are perhaps the largest institution in terms of the biological activities from in-cell to all the way to in vivo studies. And these hundreds of screens provide us opportunity to screen wide range of various natural products and to discover their biological activities. So the work in most of the cases start from field surveys. We collect information from the fields. We visit villages, towns, and interview hundreds of people to know what they are using when they're sick and what are the ways they are actually using in terms of preparation of these herbal formulations. These data then eventually come and documented in ethnobotanic databases. And then at the same time, in some of the cases, we go for extractions, biological screening, identify their structure, and wherever possible, conduct preclinical and clinical studies. In the process, we have isolated literally thousands of natural products from various plants and marine organisms as well as microorganisms. And this is a very rich source of chemical diversity, which we have developed over the years. They are not run-of-the-mill molecular bank, but in fact, they are quite diverse structures which we got from various natural sources. Now, you see, it's not trivial to isolate compounds from plants. We often think that natural product is relatively easy to conduct. The fact is that this is as challenging as always been. So it is very, very important that we really understand various stages of the isolation of natural products from complex mixtures of compound which are present in plants, roots, leaves, stem barks, and others. And for that, there are various stages of the isolation process. Of course, start from collection and authentication of plant material, drying. There are various procedures available for making sure that you do not only decompose the natural products which are present in plant matrix. And then, of course, then it comes to a very important stage of extraction. How would you extract biologically active compounds from complex matrix? And of course, this extraction and fractionation then is followed by chromatographic analysis. So there are various chromatographic techniques available. Eventually, you are able to get the pure compound. You would like to know the structure of that molecule by using spectroscopy or X-ray crystallographic techniques. We have MK in the audience. He can solve any structure of any size. And of course, then to evaluate their biological activities. And this is just a beginning because you need to work on pharmacokinetic properties. You may have to change the structures. You need to validate the biological activity by using various kinds of assays. This is the flow of the isolation. Once you get the extract, you fractionate by using various solvents. So the best method for extraction and fractionation is based on solvents. And of course, there are various solvents which are available through which you can isolate, you can extract various classes of compounds from alkaloids to saponin, from tritropine and others. So this is the simplest method of extraction. You can extract by using simple maceration, that you have a plant material, you place that plant material in various solvents, and you then filter out that extract. This is the easiest way. It's just more like extracting it in the softest possible manner, but it doesn't really give you the maximum yield, and it depends a lot on the solvent which you will use, the class of compounds which are present, the polarity of compounds, and of course the method which you will use, such as using ultrasound using sonic waves, using various solvent extraction methods, heating and reflexing and others. So a lot of these experimental conditions are extremely important when you extract the plant material, and this is largely based on the extract length of the extraction period, the solvent which you use, pH of the solvent, temperature which you optimize in order to get the maximum extract, the particle size of plant tissues. You generally try to grind them to powder so you have a maximum surface and you can extract as much biologically active compounds as possible. These are the methods which are actually used. Extraction is an extremely important step. You know, people generally fail right at the beginning because they're not able to get the maximum amount of compounds which are present in complex plant matrices. So maceration, a very simple method, infusion, decoction, reflex, Soxhlet extraction, which is a relatively improved version of reflex and was developed by a German scientist. Supercritical fluid extraction, which is state-of-the-art and is now used at the industrial level. And of course, you can use the ultrasound and microwave in order to agitate and have maximum quantities of the natural products extracted. So for instance, here you can see 3 different types of extraction. The A will represent conventional extraction in which you have plant material with a solvent, you're just agitating it mechanically, and by virtue of it, you will be able to get the extract. But you can also do that by using ultrasound-assisted methods where the sound waves are capable of improving the kinetic energy, and then by virtue of it, you will be able to get the maximum extraction because they're going to be more surface solvent extraction interaction. Microwave is also a very important technique. Of course, this is not the normal kitchen microwave. These are specialized microwave which can control temperature and at the same time have various safety features. Maceration, as I mentioned, is the simplest method. People are still using it in developed countries where the plant material is just soaked in various solvents and you filter it out. This is a gentle technique, but at the same time, yield is generally very low. This is the process which is used in maceration, but of course, far better is infusion, where you can have the solvent, the temperature is increased, and as a result of it, you get a better extraction. So in boiling water or in boiling organic solvent with the necessary safety measure, you'll be able to get the extract coming out of it. Decoction is certainly an improved version where you have you can reduce the solvent by using heating, cool and strain, and at the end of the day, you get the concentrated extract of the plant material. Reflex has been used since now over a century, and in this case, you keep reflexing the solvent, and the contact between solvent vapors and plant material eventually lead to the maximum extraction possible. And this was a version which was then improved by Franz von Soxhlet. And this method is now almost a standard method in various laboratories. And you can see that there are various two solvents. And of course, you see the solvent. And of course, you see in thimble, there is a plant material available. The temperature is increased. The vapors go through the plant material. And this extraction is reflexed. And this is continue to flow for several hours, and you get the maximum extract. You can use ultrasonic extraction method, as I mentioned. The philosophy is just very simple. maximize the interaction of the plant material with the solvent and microwave assisted extraction. The best method currently available is called supercritical fluid extraction method. The machines were really very expensive, but now the cheaper versions are available for about 5 liters. In this case, you don't really use any organic solvent. So you save money and you save environment, because organic solvents need to be disposed and is often very difficult. So what you do in this case, that you use inert gases such as carbon dioxide, compress them in particular condition, make them at a particular point, which is called supercritical. These gases are converted into solvents of different polarities depending on the pressure and temperature. And these so-called fluid or solvent are then used for extraction. The best part of it, that once you complete the extraction, you can just leave the extract out of in the environment and carbon dioxide evaporate. This is the best method. It is now used at a very large scale in the extraction of natural product. But you can also have a laboratory version of it from 1 liter to 5 liter version. And it's not that expensive because now many companies are manufacturing supercritical fluid extraction extractors. Now, once you have an extract, of course, you would fractionate in different solvents, but you need to isolate pure compound from, again, a complex mixture of compounds which you have extracted from the plant. And for that, the easiest method was the thin layer chromatography. This was the classical method. So column and thin layer chromatography were actually used, and these are solid liquid interactions depending on the affinity of compounds. For both solid phase and mobile phase, you can separate these compounds one after the other. And if you're lucky, you would be able to get pure compounds from the column. If you're not, you perhaps need to use various other methods. But the basic principle of chromatography is the use of mobile phase and a stationary phase. And you have a whole range of choices for a stationary phase. Many stationary phases have been developed. So chromatography is used for separation of compound, which is very important, purification of substances, identification of compounds. If you're coupled it with the spectroscopic technique called hyphenated technique, you can use them for quantitative and qualitative analysis. And of course, depending on the quantity of and nature of analyte, you use various chromatographic techniques. So you can use from gas chromatography to HPLC, ion exchange, and size exclusion chromatography. Thin layer chromatography is the simplest one, which are still being used. Column chromatography, with the choices of now over 24 stationary phase commercially available, from simple silica gel all the way to various Cefidex and others. You can use various chromatographic techniques and isolate compound both on the basis of their polarity, the light dissolved like the basic fundamental principle of dissolution, two sizes which can be used to separate compounds of different sizes. So ion exchange, you may have compounds with different charges, particularly in the field of biochemistry, where people are working on various proteins and peptides. Ion exchange chromatography is used. Size exclusion chromatography is the second phase, where you have already fractionated your compound on the basis of polarity, and you're not really sure whether you would be able to separate them further by using simple solvent polarity-based column chromatography. In that case, you use size exclusion chromatography, for which the various stationary phases have been developed, and you can separate them on the basis of sizes, not the polarity. There are various versions of size exclusion chromatography, but in all such cases, kindly remember that you don't change the mobile phase. You keep using the same mobile phase, which are various organic solvents. The only thing which is functioning in this case is the stationary phase, which is specially designed to hold certain molecules of certain sizes. And this has been a very effective way. You can use these Saphadites and the different types of metrics which are actually developed, and you can use them for several cycles. And then, of course, you also have a choice of using gas chromatography in case of non-polar compounds, volatile compound. These are the compounds which you would like to isolate for pharmaceutical purposes, for aromatherapy, for fragrances, and others. And gas chromatography, the only key difference is that you are not only using the solvents, organic solvent. Instead of that, you are using various inert gases. And your gas, your mobile phase, which is a gas, as well as your compound, they are in volatile. So it is an extremely robust technique, gas chromatography for volatile aromatic non-polar compound, particularly suitable for terpenoids and non-polar organic compounds. But if you would wish to isolate polar compound, then you have the choice of HPLC. HPLC is now almost a standard method for the isolation of polar constituents. And again, in this case, it is just it's an automated version of column chromatography, but with the added advantage that you can use various pressures. And the theoretical plate, the compactness of the column enable you to isolate compound with great precision. And the process is very rapid. You have the choice of various solvents, and you can mix those solvents in gradient manners. And by doing this, this has almost become an online isolation where you inject your sample for analytical as well as preparatory purposes. And by using various columns, you would be able to isolate compound. Now, a better version of this available since last 15 years is called ultra performance liquid chromatography. And this is an improved version of HPLC. Of course, with added cost, and roughly about 9 times more effective in terms of isolating minor constituents in a very cost-effective manner, because you do not really need to use various solvents. So these are UPLC or ultra performance liquid chromatography. They're high resolution, high speed, 9 times faster than HPLC, quick and easy to handle. And of course, you can really complete the process in few minutes rather than several hours. So there are various techniques available, but perhaps the most important technique which was developed and named by Hirschfeld was hyphenated techniques. What was done was that the two techniques were joined together. One is chromatographic technique and other is spectroscopic technique. Chromatographic technique can be anything like from HPLC to GC and others. And then spectroscopic techniques can be such as mass spectrometry or NMR or IR. The objective of hyphenated technique is the online rapid isolation and identification of compound. The chromatographic techniques help you to identify, to isolate compound from the complex matrix, and the spectroscopic technique help you to identify that by using various techniques such as mass spectrometry, NMR, and others. Now, there are a range of such techniques available, and the most common one is GCMS. And of course, HPLC-MS, which is called LC-MS, MS-MS, standard mass spectrometry, with various kinds of detectors. LC-NMR is also commercially available, which is a bit of a challenging because LC requires lots of solvents. So interfacing is always a major challenge when you combine a chromatographic technique, which is a very different animal, as compared to mass spectroscopic techniques, but people have successfully done it, and many of these equipments are available. They are particularly interesting and important because not only that they have the advantage of fast and accurate analysis, high degree of automation, you can have high sample throughput, better reproducibility, and of course, almost contamination-free, but also indeed application of natural products. You know, we keep working on plants and we keep isolating known compounds after years of efforts. But now with the advent of hyphenetic technique, it become easier that you can identify compounds which are already being known. So it is a standard tool for deed application of natural products, and it saves a lot of time and effort which are actually which was classically used in the isolation of the same compound again and again. This is a schematic of hyphenated techniques and how one can do online isolation and identification. So you can see that sample is injected in any of the chromatographic techniques, such as liquid chromatography or gas chromatography or even ion exchange chromatography. And of course, then by using various kinds of detectors, there are tons of different detectors available. You would be able to see whether you are getting pure compounds, and then by using standard splitters, you can split the samples coming out of the column, which goes into 96 well plate, and you can carry out almost automatically these biological studies, which are now standard methods, particularly the biochemical assays against different enzymes and proteins. But at the same time, you would also be able to take them directly to LC-NMR, LC-MS, LC-NMR-MS, and you get a lot of data for the identification of compound. And of course, many of these libraries are available for natural products, and some of them are available in public domain, so you don't need to pay them. Some of them are extremely expensive, but in any case, you would be able to identify compound, majority of them, if they are known and present in databases. And this makes this the entire exercise of natural product chemistry relatively easy and time efficient. Now, of course, once you have pure compound in hand, you go for the biological activity. And in the last 15 minutes of the time assigned to me, I will discuss the example of a work, one example of a work. And this is against the Leishmaniasis. We know that entities are very common. I don't need to repeat what has been said earlier, that there are many diseases. They are neglected by virtue of that being, these are diseases of poverty, and not much resources are available for research and development. And unfortunately, they are neglected in all different spheres. So you can see that majority of these diseases are prevalent in tropical and subtropical regions, which are also global south, with poverty very common. And many of these countries, you can see that they have more than one tropical disease, neglected tropical diseases, and they are because of various infectious agents from elementals to protozoan parasites, fungi to bacteria and viruses. And Leishmaniasis is very important, and particularly cutaneous Leishmaniasis among neglected tropical diseases. I remember what Paul said, that they are even more neglected. So they are neglected, super neglected diseases, and particularly among various forms of leishmaniasis, cutaneous leishmaniasis is very common, prevalent, increasing in number, prevalence is increasing, and with the climate change, it is expanding into north and south, and becoming increasingly common, particularly with the co-infection of other diseases. It is because of many poverty-related problems, and the treatments have been discussed. I'm sure that you know that the current treatments, majority of them are repurposed. They're not designed for leishmaniasis, so they're not only made for leishmaniasis, but being successfully used for the treatment. Unfortunately, many of them are expensive and not so good. This is a disease which is zoonotic in nature. In Pakistan, the entire country is affected. It's a big country with a population of 240 million, the fifth largest country, and as has been mentioned, that the hospitals are flooded during the seasons of leishmaniasis between October to March and April, where we see lots of these cases coming, and outbreaks are reported from all over the country. And often WHO has to declare emergency of airlifting medicines from different countries to bring them to Pakistan. And in this context, we have been working since last decade on various plants. And based on ethnobotanic survey, we have actually identified 4 plants. I'm just going to give you the example of one of it. And we have successfully isolated anti-leishmanial compound from 3 of them. And in 2 of them, we were able to take them to preclinical stage. And since they are already used in field, we know that their efficacy is validated. So let me take you through the work which we have conducted on Physalis minima, and I'll give you the context of how this work was conducted. So anti-leishmanial assay, very medium throughput assay in which various leishmanial strains. Some are maintain a range of leishmanial strains in her laboratory, and some of them are clinical isolates, some of them were purchased from standard culture collection. So we have a whole range of different strains, and we are also able to develop resistance in them. So we have both clinically resistant and laboratory-based resistant strains of various leishmanial strains. And the work which I'm going to present to you is on a plant called Physalis minima, which is a weed. Plenty of this plant is available. And we started working on this plant since last 7 years, and were able to identify compounds from this plant. But of course, we have conducted conducted clinical trials, topical formulation for cutaneous leishmaniasis. Recently, we are working closely with the Durham group to identify which strains are common. We know that the strain is actually changing. It used to be Leishmania major, but it's now co-infection with Leishmania tropica as well. So Physalis is a medicinal plant. It is used in a variety of different therapeutic indication. We were able to identify compounds and conducted clinical trials on 80% methanolic extract of the aerial part of Physalis minima. We have done everything from ethnobotanic survey to clinical trials of the methanolic extract, and then to identify compounds. What is missing was the mechanism part, which is certainly something which we would be looking forward to have a very active collaboration. We have already developed some good partnership and linkages with leading groups in the world. So from isolation, till the structural elucidation and then further structural transformation by using biocatalytic and chemical methods. We created a lot of chemical space and were able to demonstrate that some of these compounds isolated from the plant were active. So clinical trials were conducted on some 100 patients in the Leprosy Center, Larkana. And in the season, we are able to contribute some of these formulations for the field trials. And we also got US patent, so this is already intellectual right protected. This is the extraction. As you can see that from methanolic extract to various solvent-based extractions, you can better appreciate that in the context of the method which I have presented to you. Various chromatographic techniques were used to fractionate various classes of compounds from very non-polar to polar compounds. And of course, the activity was compared with 3 standard drugs against 2 major strains, Leishmania major and Leishmania tropica, amphotericin B, pentamidine, miltefosine. And the comparison has given us the idea of the activity of the constituent which we have isolated. So fractionation was done in a very logical manner. We made sure that every extract is checked for the activity against the promastigotes of these 2 Leishmanian strains. And as you can see that compounds were isolated. So you can see they are fairly complex. They are withanolite, steroidal lactones filled with lots of functionalities. Not so easy to synthesize, but luckily many of them are available in sufficient quantities by using the method which we have developed from plant. So you can see that some of them were found to be more active than the standard drugs. And these are, some of them were as active as amphotericin B or miltefosine and all that. And among them, the hydroxyphysallene, the first one was the most active one, but the one which we got in largest quantity was physallene H. And we have then modified the structure by using microbial transformation. So these are some other examples of the molecule which we have isolated. Of course, not every compound was active because we have isolated large number of compounds from Physalis minima and then identify which one of them was active in promastigotes. We have then moved on to, again, this was the result of this network GCRF NTD that we were able to introduce several new assays. And now we are able to target a mastigos stage of parasite in cell lines and to check the activity within the cells. And as you know that mastigos are the important component of the life cycle, which actually infect humans. And for that, we have actually used a cell line called Crown. It is called Crown J774. The cell line was used to grow macrophages in large quantities. These macrophages were then screened by using atomic force microscopy and screening electron microscopy. Various compounds were screened against the mastigotes in cell lines, and we found that these compounds were active to mastigot stages also. And then we looked into the morphology of these mastigotes. We have done simple theoretical studies by docking these compounds against various important enzymes, which are important in metabolic cycle of Leishmania strain. And for that, these enzymes were targeted glucose 6-phosphate isomerase and also myristoyl transferase. These docking studies were also conducted against phosphoglucose isomerase, and now we have some indication that which particular compound is predicted to be docked against various enzymes. So this has been done. This is purely in silico studies. We have not really gone to biochemical assays so far. We have also, in the last few minutes, that we have developed miltefosine resistant strain of Leishmania tropica and checked our compounds, both natural and their synthetic derivatives, and was able to identify that some of these compounds were active against the resistant strains also. So this is the kind of work which we have conducted. Of course, a lot of studies are being conducted even now, and many new compounds have been identified which were active. So these are the results of our work on resistant strains. And as you can see that some of them were as active as amphotericin. Even on resistant strains, they were even more active. I'd like to acknowledge, number one, Summer has been the key investigator in this particular study. I had the privilege of supervising her work when she was my student, and now we work as a colleague. And I'm so delighted to see her intellectual capabilities nurturing into the next phase. Of course, there are many other students who have contributed along with the Summer, and they are the part of our team. Wherever we were very fortunate of developing excellent network with Durham team and with colleagues in 5 other countries. So this is the work which is the result of a lot of interdisciplinary and cross-disciplinary interaction and collaboration. I'd like to thank you for listening, and my apologies for this technical glitches, which was, I'm not really sure where they arise. And thank you very much for hosting this important event. We intend to see some of you coming to Pakistan and working together with our scientists. Thank you.
Thank you, Professor Iqbal. It's a very comprehensive presentation. I think those of us who are interested in working on national products in an attempt to look for lead molecule from natural sources, this is a very eye-opening presentation. I wouldn't attempt to summarize. I think in the interest of time, I would rather briefly mention what he has said, starting from extraction, going through isolation, using different techniques, including macerations, for example, decoction and the likes, then getting to characterization and subjecting to test for anti-Leishmanial activity, and going through both preclinical, clinical developments. It was really wonderful, Prof. Iqbal. Thank you so much indeed for your nice presentation. Thank you, sir. I'm not sure whether we have time for questions. Unfortunately, in the program, there is no slot for discussion, which was wrong. But I wouldn't leave you astray without at least giving one chance, both from the online as well as from the in-person audience. I know there are a lot of chats, a lot of questions and comments from the online attendees, which we may not be able to accommodate all, but I'll give one chance for those who are interested to ask questions or leave comments anyway. Let me start then from the face-to-face audience. Just one chance only, don't miss it. Okay, we don't have time to go through. So any burning issue you have, okay, Dr. Asbab. Wait. I wish you could come there and close. Dr. Tekmak. Please come here. Okay? Yeah.
Okay, thanks so much, Prof. Yasu, for giving the opportunity. I appreciate all the presenters for educative and highly stimulating presentations. My question goes to Professor Asrat. I'm very curious to know whether is it really a zoonotic disease because it affects only the dogs? Does it affect other such as cattle or? And the other my curiosity is there is colasm, they call it colasm by the veterinarian entomologist. It's very common, especially in Matama area and the Gambela. Is it really sandfly? Because if it is sandfly, they have a sort of controlling program so that we can collaborate with Animal Health Institute. They have a research team working on colasm. I don't know. They may also be familiar with this Leishmaniasis. That's my question. Thanks so much.
Thank you, Dr. Asfar, for the questions. Please make sure to jot down the question. Okay. Maybe from the online attendees, is there any question which you want to say it out? I don't see any hand raised. You have it. Is there anyone? Huh? No questions. Okay. Okay. Uh-huh. Yes, because she's a lady. Okay. Okay, let's give her a chance then. Han. Okay. Han, you must be lucky. You have been chosen from the online attendees. Yes, to raise a question. It's Moses or Han? Moses. Oh, I'm sorry. Okay. Moses, you have raised your hand. Could you please?
Thank you, Prof, for the opportunity. Yeah, this is Sanya Moses, coming live from Nairobi, Kenya. I'm at the University of Basic Science, Technology and Innovation, that's PAUSTI, hosted at Jomo Kenyatta University. I'm a master's student pursuing my MSc in molecular biology and biotechnology. I would like to thank the presenters for the powerful presentations that have been quite informative. Yeah, it is good in creating awareness and building capacity. Personally, I'm undertaking a research project titled Development of a real-time multiplex PCR kit for detection of Leishmania and HIV co-infections. Yeah, so this training has been so helpful to me. Yeah, and I look forward to working with all of us. Thank you so much. I remain Moses Sanya at PAUSTI.
Thank you, Moses. Whom do you like to address the question to then? There were 3 presenters, okay? Whom do you prefer to answer your question? Moses. Can you hear me?
Sorry, it was not a question per se.
Yeah.
I was just appreciating the opportunity to attend the training. Yes.
I see. So yours is comments then. Thank you so much indeed. Okay. Professor, could you come here in front so that online attendees could also listen to you. Okay.
Thank you. So the question is Leishmaniasis really zoonotic? Okay, so that was one question, and the other one was about. Yeah, so he mentioned colasm. I think colasm is tsetse fly or not? We have Marcos here, so colasm is not really related to the sandflies, so that solves the problem. So, but again, sandflies and veterinary problems, I haven't seen any association yet. In terms of being, veterinary problem, Leishmaniasis being a veterinary problem and transmission by sandflies, I haven't come across that kind of information. But the main question is really, is Leishmaniasis zoonosis? And just imagine Leishmania atropica, we have hyraxis. So in the in Ethiopia and also in Kenya. So a couple of species of hyraxis. Again, parasites isolated, Leishmania tropica isolated from hyraxis. Dogs, obviously, phantom, but in Sudan, they also isolated Leishmania donovani, from dogs. But wild animals as well, like mongooses, Egyptian mongoose, dwarf mongoose, and so on. And cutaneous leishmaniasis in Americas is largely zoonotic, largely in the jungle, people get infected in the jungle. So yeah, India, and still, their visceral leishmaniasis is considered to be, anthropogenic, even though some people still doubt or consider that there might be a natural host. But the problem in India is it's a high incidence, it's all the time. And so in that scenario, it's always possible that it is transmitted man to man. And the sandflies bite on humans as well as animals. In fact, they bite a lot on animals, more than humans. For instance, phlebotomous orientalis, which transmits diseases, they bite on cattle, so 90%. So, when you took into account all these evidences, yes, indeed, it is a zoonotic disease. So that's what I can say at the moment.
Thank you, Professor Rat. Iqbal, do you have any comment whether Leishmaniasis is zoonotic or not?
This is a question for me.
If you want to comment, it's not mandatory.
Certainly, I'm absolutely not qualified to answer this, to be honest, because this is something which is related to more epidemiology and people who understand that. As far as we know, the literature in Pakistan and as also in Afghanistan, Iran, and West Asia, there are a lot of reports by Japanese researchers who have isolated leishmanial strain from livestock, cattle, and also from domestic pets such as dog and cats. So Japanese researchers have actually worked on various strains of sandfly, and they were able to isolate leishmanial strain from some livestock also, which are commonly raised in this part of the world.
Thank you, Professor Iqbal. Professor Paul, do you have anything to say? You want to add? I don't know.
I don't think I have it.
You don't have anything? Okay, thank you. I'm afraid we don't have time. We are just pushed by the time constraint. I hope you understand. Okay. All right, thank you. Ecolasm, I think, does it bring about trypanosomiasis instead? Yes, it is. Yeah, it's not Leishmaniasis. Okay. All right. So, come to the end of the morning session. I do have great appreciation for all the presenters, starting from those who have given us their remarks. I think they deserve Why do you keep quiet? They deserve what? Thank you. I would also like to thank all the audience for active participation. Had it not been for time constraints, could have entertained a lot of questions and comments. I know there are a lot of comments on the chat box, but I have been pushed by the time, then I would not be able just to give chance for these comments to be surfaced, unfortunately. So your active participation and your patience starting from the morning 9 up to now, I know your stomach is getting empty, but in spite of this, you still bear with us and you have chosen you have shown your patience, and you also deserve a round of applause. Finally, I would like to thank the organizers for making this to happen. I would like to thank Dr. Fozia for the link she created. And I'd like to thank CDT Africa, in particular, Professor Abeba, for making it happen as well. So could you please appreciate them as well? So with this, we'll go for lunch. We'll have afternoon session. That's purely lab work. Only those who have been selected for the laboratory are invited. And tomorrow, we shall gather together once again in the morning for other presentations. I believe you would enjoy them as well. So you are cordially invited to attend tomorrow's session too. Thank you so much indeed.
Thank you very much. Thank you.
Thank you.
Wow. No, okay. That's center we can much other. Oh, yeah, no. That's scary. Yes, you know, it's በኋላ this little bit, that's about chunk. What's the sense? መጀመሪያ ይህን አጥፎ ነው። ስለዚህ ይህን ያቀው ይህን ያቀው ስለዚህ አይ ለሞክር ሳይሆን ማድረግ አለባቸው። Okay, excuse me, there is one announcement here. Sorry for the change of. Instead of going directly to Korambetsa Specialist Hospital for the practical, since there is a plenary lecture to be given by Professor Asrat, which could help students understand how they could move on with the practical sessions. Then instead of doing it there, because as you know, today is holiday, there is no one who can help us there. So it might be better, just after lunch, you may come back here and then you could get the lecture proper. Thereafter, you would go directly to Dash for the lab practical. This is applicable and only for those. By the way, the lecture is free for all. Okay. Yeah, but the practical is only for those selected. Thank you, because of space problem.
It's only the pre-legal with the comfort, we haven't got. We also haven't gotten the third one. The previous indicates not 3 years ago, because so why did they celebrate? So why don't you have tomorrow we don't have. Well, you can't after tomorrow we have products for take out for free parts. That's okay. Okay, one day next tomorrow, yeah. I don't know. Yeah, for living long.
We managed to go with the chats.
Yes, yes. First yes, you have but our class must have recorded it.
Yes. On that.
I think they kind of the move that we have it download on the chat.